thank you for this kind invitation, and I hope you that you can understand what I'm talking about. I will try to be very didactic, and I will talk you about genetic tools to study brain function, and if there is some time left, how we can focus the study on our main research subject, which is the endocannabinoid system. You can raise your hands or talk to me, and please really feel free to interrupt me at any moment of my talk. So as I said, the main body of the talk will be the overview of genetic techniques used to study neuronal function. And then I will give you an overview of the physiology of the endocannabinoid system. And of course, if there is still some time, the difference of neuroanatomical and functional heterogeneity of CB1 signaling that we can discover, especially thanks to genetics technique. So, as we know, studying the brain, it's a very complex issue, because for all the type of studies we do in biological system, we go always from functions to the molecules involved, or vice versa. And for this, we can use two approaches. We can use what we call a top-down approach. So we start from a function, and we go down to see which are the molecules involved in this function. Or we can do the opposite. We can use a bottom-up approach, going from given molecules to understand the function in which they are involved. Now, if we take a simple organ like the liver, where the main cell type is hepatocytes, which they do everything, we can see that often the two ways of working, they can cross each other. So we can go from function to molecules, or vice versa, because it's a very simple system. But if we take the brain, the level of complexity is huge, and the two approaches, they do not cross, so we have to choose one. And I give you an overview of the two of them. So first, the big question of the lecture today is how can we use molecular biology techniques to learn about brain functions and behavior eventually? And can we obtain more direct information that links molecular biology to behavior? And what techniques can be used to do this? So as I said, there are two general approaches. From neuronal function to molecular biology, or from molecular biology to neuronal functions. For the first approach, it just consists mostly in the analysis of phenotypes in inbred mouse strain, which are selected for a typical behavior, in mutant animals that can be naturally occurring or artificially induced, but I'm talking here about random mutant animals, not transgenic. Or it may be human population, families, twin, restricted communities, etc. And the idea is to identify, to run genetic studies to identify the genes involved in the phenotype. For example, the quantitative trait loci analysis. I want to just do a little disclaimer before that I will use a lot the words genotype and phenotype. Do you want me to refresh what these terms means, or is more or less clear to everybody?
It's clear, I think.
It's clear. So genotype is what the genes are, and phenotype is what is caused by these genes. Okay. So we do what we call a project-driven approach. We go from a function or to a malfunction, which can be a disease, to the genes involved. And the advantages of doing this is that we can have important information of genetic basis of the disease. This is how Huntington disease was discovered, how some mutations that are linked to Alzheimer's can be used in humans. So this can really bring a complete genetic analysis of complex disease like schizophrenia. And but however, has disadvantages that is very long. It needs a huge amount of data to make some correlation, and necessarily needs a second step hypothesis, meaning that if you identify a gene involved in a disease, then you need to study what this gene and this product, however, or through the protein, does. From the other approach, from the molecular biology to the neural function, is what we call the hypothesis-driven approach. So we go from a genes to its function or its malfunction when it's missed or it's tampered with. And there are various methods to introduce novel genetic materials and or to modify the function of existing genes in brain of living animals. And this is what I will talk about today. there are transgenic approaches, what we call the gene targeting, which is based on the homologous recombination. Local injection, especially now in neuroscience, is what is going most. It's viral vectors, but we can also use small interference RNA or antisense oligonucleotides, which is all the outdated technique that is not very much used anymore. And we can also have combinations of all of them. And the advantages of doing this kind of study is that can provide detailed information of single genes or proteins. It can allow dissecting different functions of the neuronal system, and it's funny. And the disadvantages with this is that it's expensive, because especially with studies in animals, and cannot be used in humans, because you need animal models from the neuronal function. And although some people are getting closer, it's very difficult to have nowadays a schizophrenic mouse. So let's start a little bit with the technical part, the transgenic approaches. What consists the transgenic approach? It consists of injecting linearized DNA into the pronucleus of fertilized oocytes. So this can sound very complicated sentence, but I will try to facilitate it. So we know that an oocyte, when it's been fecundated by a sperm, it has the two pronuclei that then we will fuse into unique nucleus. So what we do, we in vitro fecundate oocytes with sperms, and when we arrive at the level of the pronucleus, with a micropipette, we can inject a piece of DNA, linearized DNA, into one of the pronuclei. Normally, we use the male pronuclear, which is actually bigger and easier to see. Don't ask me why, people never understood this, but we know that it's the male pronuclei. So what will happen? There will be a random integration of this piece of DNA into the genome. Of course, random will not be just the position, but also the number of copies integrated. And what we do, we take a female mouse, we induce a pseudopregnant state with hormones, and then we reimplant these oocytes into these females. And each oocyte that has been manipulated, because we do this in several ones, will generate a mouse that will be called founder, and will be a transgenic founder. What can we do with this transgenesis? We can overexpress endogenous protein in specific cell population. And on the same hand, we can overexpress mutant protein that can be dominant negatively, constitutively active, dysregulated, fuses to other protein. Or even we can express exogenous protein in specific cell population, of course. And this you will understand why later. And to do this, we need our artificial piece of DNA, which in jargon it's called construct, that contains the promoter or other regulatory sequences to start the transcription, the genes to be expressed, which can be endogenous protein to be expressed, mutant protein, or even exogenous protein, and the polyadenylation signal to stop the transcription and restrict the transcription only in the gene we want to express. And I mean, you can guess between the three of these, apart the gene to be expressed, what is the more important thing? Is the promoter and the other regulatory sequence, which they give the specificity of expression. So what are regulatory sequences? Are sequence of DNA that are called transcription machinery. It's the sequence of DNA where transcription factor binds to transcribe some genes. And they are mostly constituted by the TATA box and the famous Kozak sequences. And this constitutes the minimal promoters that each gene has to have to be transcribed. But we have other regulatory sequences which can boost the transcription, for example, in different tissue, block it in another tissue, which are called enhancers and repressors. So the minimal promoter, it's a short sequence, just little than 10 kilobases of DNA that are necessary for transcription specific genes. It can be cell type specific. It can be ubiquitous, which means that it's expressed in every cell of the body. It can be bidirectional because now there are synthetic promoters that they can express two genes from the same construct. It can be strong. Normally, these are viral promoters. It can be weak, according to really what we want to do. And as I said, it can be of different origin. Normal constitutive strong promoters are viral. Cell type specific promoters are mostly animal. And the advantages of using a minimal promoter is that it's very easy to use, because the more DNA you manipulate, the more difficult it gives. It can have a small dimension and can be generated just by PCR amplification. And it can allow, since it's smaller, several integration of several copies in the transgene, and this can modulate the expression. And sometimes it's sufficient to give good specificity of expression. However, the disadvantages of this is that you can have different expression patterns between different founders. So you do a lot of work of analysis to find the right founder. And you can have different level of expression in different mice at different ages, because you don't know. This can depends where the piece of DNA has been integrated, and also in different tissues. That is what we call the mosaicism. However, in neuroscience, this technique has been used, and in 2000 it was published the first transgenic mouse that is nowadays used in neuroscience, that are called the gene mice, meaning GABAergic interneuron mice. So what they did to visualize these neurons, they basically wanted to express an exogenous protein that is called GFP, that is enhanced green fluorescent protein, that is a protein of jellyfish that upon UV shines green light. So what they did, they made a construct with a small sequence of the mouse GAD1 gene, the promoter. So they cloned by PCR a small piece of the GAD gene, and behind they put GFP, so promoter, the gene to be expressed, the polyadenylation signal. They made the injection in oocytes. They found the right founder, which under a fluorescent microscope in slice has all the interneurons labeled in green. However, to overcome the limitation that I told you before, we can use even more specific regulatory sequence. And what we use BAC. BAC stands for bacterial artificial chromosomes, which is a library that has been created first for human and mice. That is a library that contains the whole genomes in form of bacterial artificial chromosomes. So bacteria, they take pieces of human genome and they make small chromosomes. They can be up to 200,000 base pairs, and they contain given genes. So we can choose the BAC that contains the given gene we want with the promoter, and we can take this promoter in the BAC to express our gene of interest. And we do always the injection in oocytes and the selection, etc. And the advantages is that using a BAC, we have very high specificity of expression, because most, if not all, the regulatory sequence of a gene, they are present, and the level of expression for this are similar to the ones of endogenous protein. However, the disadvantage for the BAC is that they are very, very difficult to manipulate. Now they are kids, but still, because they're very big pieces of DNA. And there is a danger to introduce unwanted genes, because in a BAC, you can have several genes. And sometimes the level of expression cannot be sufficient for certain purpose, because we need to boost the activity of endogenous promoters. However, as well, I'm giving you the first example of a transgenic mouse that is still used in neuroscience. Is a mouse that targets the expression of Cre recombinase, which is a bacterial protein, and I will come on this later, in principal projecting neurons, so CAM kinase 2 alpha. So what they did in this study, they took a BAC which contained the CAM kinase 2 alpha gene and 50 kilobase of its promoter, so quite a lot of DNA of regulatory sequences. And before the starting of the CAM kinase 2 alpha, they inserted Cre with the GFP cassette. So what they did when they inserted this, and when they characterized the transgenic mouse, we can see that an immuno against Cre, so detection of Cre with antibodies here in brown, you can see that respects the pattern of principal glutamatergic projecting neurons, especially here in the hippocampus, which the immunoreactivity is localized only here, where you have normally glutamatergic cells. So now what I explained you is just transgenic mice that they do overexpress something, even exogenous, all the time. But we can also induce the expression of a transgene in specific neuron and the specific time points by using the inducible system. This is a system we borrowed from bacteria, because we know that bacteria, when they are under antibiotics, they change completely their physiology. And one way is to change the expression of genes. So they have a transcriptional tetracycline, which is an antibiotic regulated system. The idea to borrow this system from bacteria is to make a transgenic mouse which has a cell type specific promoter which express the TET transactivator. This is a transcription factor that is bound by tetracycline. And on the other hand, we can have the DNA sequence called 3 TET responsive elements, which will be bound by this transactivator, a strong ubiquitous promoter to boost the transcription, and the gene of interest, the transgenic that we want finally to express. So what will happen? That in certain cells, it will be produced, given to this promoter, will be produced the TET transactivator, which will bind to the 3 and will trigger the expression of the gene of interest. However, when we give the tetracycline will bind to 3 will detach from the DNA, and the expression of gene of interest will be stopped. And this can be triggered again by removing doxycycline. I must tell you that nowadays, doxycycline is very easy because it's administered normally in the food of the mice, so you don't need to make injections. So as you can see, this system is very optimized. And this is called the TET system. So when you give tetracycline, you stop the transcription of your transgene. Now, and I will give you an example, it exists only the TET-on system that you need doxycycline to boost the expression. And this is an example from a paper, very nice, in 2001, that they wanted to study the calcineurin inhibitor, which is a peptide, and they did an inducible overexpression. So what they did a transgenic mouse that under the CAM kinase promoter, it's expressing the TRE, the TET transactivator elements with the TET-on way, and under the TRE, they put the calcineurin inhibitor. And when they do a Western blotting for this, they can see that in the hippocampus and in the cortex, the control mice, no expression of the transgene, mutant mice, nothing. Mutant mice with doxycycline, they do have expression. And when they remove doxycycline, the expression is not there anymore. And you can see it's mostly glutamatergic. Same thing in the cortex. And then in hippocampus and cortex, when they test the activity of calcineurin, since they are expressing a calcineurin inhibitor, they can see that only the mutant mice with doxycycline, they drop the activity of calcineurin, and they can recover it when they put back doxycycline, when they remove the doxycycline. So, and this was studied in this study, if you are interested to read it, actually this manipulation was interfering with the formation of fear memories. This is a study from the lab of Isabel Mansour. Okay. Now on, we said how we can do a transgenic mice that express something more respect to his genes. But what about if we want to study the function of genes that are already there in mice? We can do what we call the gene targeting, which is the fancy name for a very physiological process that is called homologous recombination. So homologous recombination is the process by which two homologous chromosomes can exchange some of their portion during the meiosis. You know, the meiosis is the process that is happening for the gametes formation. So the way through which we can introduce genetic variability in our progeny is that during the meiosis, so generating gametes, our chromosomes are exchanging some of their portion given to some complementarity. This was known since more than 100 years, but what is new about it is that it can work with exogenous DNA. So a piece of exogenous DNA can exchange some of its portion with our chromosomes, and it can work with embryonic stem cells. So not only during the meiosis, but also during mitosis, and especially in embryonic stem cells. So what can we do with the homologous recombination? We can substitute endogenous genes with other genes. So we can do what we call the knock-in. We can delete endogenous genes in all cells of the body. So we can do a knockout. Or we can delete endogenous genes in specific cell and tissue, or at specific time points by what is called the conditional knockout. And I will explain all of this to you. So what do we need for homologous recombination and its fancy name gene targeting? We need embryonic stem cells. They need to be good embryonic stem cells. So they have to give what we call the germline transmission, meaning that they are capable to generate an individuals. we need a targeting construct, we need patience, and most of all, we need luck. So what are embryonic stem cells? I don't know if you have some notion of embryogenesis. However, what we can see is that when the oocyte is fecundated by the sperm, we will have the two pronuclei which will fuse in one unique nuclei, and it will start a series of cell division which will generate a mass of cells that is called the morula, which is constituted by totipotent cells. At a certain point, these totipotent cells will commit in two directions. One direction will generate the blastocyst, which then will generate the placenta, the chorionic villi, and all the embryonic annexes. And a little part of these cells will generate the inner cell mass, which are pluripotent, that are the embryonic stem cells, which will then generate the three embryonic sheaths, and then from the three embryonic sheaths, the entire individuals. So these are embryonic stem cells. What we can do with these embryonic stem cells, at least from mouse 1, which is allowed, they can be cultured in vitro. In fact, what you do, you take a feeder cells layer. So these are fibroblasts that have been killed by gamma radiation, and they can provide the substrate and the nutrients for embryonic stem cells to grow, and they grow in chunks. As you see, they grow as an inner cell mass. But this inner cell mass, it can be dissociated, and it can be implanted in another blastocyst. And what will happen when you implant this in another blastocyst? You can give rise to chimeric pups. So to make the story short, and this way I'll explain you why, as you can see here, there is something weird. We have a white mother and different type of pups. We have brown pups, black pups, and brown pups with black spots. Why? Because we use this code color. Because what we do, the embryonic stem cells that we normally use are derived from a mouse strain that is called 129, which are agouti, so are brown, and it's the dominant character. The blastocyst, instead, is normally taken by C57 black 6 mice, which is a mouse strain that is black and is a recessive character. So what we have done a chimera. We have been injecting transgenic modified embryonic stem cells from 129 strain into a blastocyst of a black 6 mouse. So you have a chimera, you have a mixed individuals, brown fur with black spots. If you take one of these individuals and you cross with black 6 and the progeny, so black 6 which are recessive, so black 6, if the progeny is getting agouti, it means that the gametes, they've been generated by the embryonic stem cells that you've been injecting, and not but the one that were residing in the blastocyst. And this will mean that you got what you hope, the germline transmission. And I want to come back on this point, why I said luck, because this normally happens in 1% of the cases when you want to generate a knockout. So I said what is about embryonic stem cells, but then we need also a targeting construct. Let's imagine that we know the whole genome of the mice. We want to target this PIPO gene. And we know that before and after this PIPO gene, we have a given sequence of DNA that we know. So what we can do, we can clone these sequences and insert it inside a neomycin resistance cassette. And then we come on this point. When we put this construct together in the cells, we will have recombination. So if we transfer this construct, it will recombine. One cell will have recombination. Then we select these cells by applying neomycin, which is an antibiotic that kills embryonic eukaryotic cells. All cells will die, but this one, which will reproduce. And then this chunk of cells will be dissociated, injected into a blastocyst. And if we have our chimeras and we have germline transmission, we will have a knockout for the PIPO gene, which have been substituted by a neomycin resistance cassette. However, if together with the neomycin cassette, we put a modified version of Pippo or another gene, we will have a knock-in. So the principle is the same. It's just that in one case, we remove a gene and that's it. In the other case, we remove the gene and we replace with another one. So just to give you some overview of this method, the global knockout or knock-in has some advantages, of course, that you can delete or replace any gene of interest. This can provide detailed information on the role of a specific gene, and the knock-in can allow to substitute a gene with another gene, which will have exactly the same expression pattern of the endogenous gene. So maybe imagine you want to mutate just one base in one gene. With the knock-in, you can do. But of course, you have to remember that the mum is always important, also in science, because you are looking for the function of a gene in a single animal. And the environment of development is very important. You can have the uterus, the birth, the milk, the maternal behavior. If the mother is knocked out, all these parameters might be altered. And often some people that were doing selected breeding by the breeding of wild-type mice and the breeding of knockout mice, they were finding phenotypes in pups that might be due to a secondary effect. So what you do to avoid this is that you use litter-mate controls. You take mothers and fathers that are heterozygous from the gene you want to study. So they have one knockout copy and one wild-type copy. And from this, you will have heterozygous to keep your colony. You have wild-type and knockout, which are brothers. They came from the same mother. So the only difference between the two of them will be the genotype. Of course, global knockout and knock-in has disadvantages. The first one is that deletion could be lethal. You have no development because you need this gene. You can have compensation because, I mean, a knockout is a knockout since the embryogenesis. And the lack of one gene can be compensated by another gene. And sometimes the deletions can generate aberrant proteins. These are most technical difficulties, and there is also a theoretical disadvantages. That is that a global knock-in knockout cannot give any temporal or spatial information on the role of a gene, when and where the gene might be important for certain functions. So, for example, I want to give you this example of this was the same group that tried to generate a knockout of CREB, that is a transcription factor. The first thing it happened to them was that in the knockout, there was actually an overexpression of CREB because, as you can see here in immunohistochemistry, because actually there was CREM, which is a similar gene that took over the same function. And the other time, they had an aberrant protein because they just targeted neomycin cassette before one exon. This exon was skipped, and they generated an alternative protein. So they were not really lucky. However, just to come of so this was a technical difficulty. Coming to the theoretical disadvantage, which is the temporal and spatial information, what we can do is a conditional knockout. And this is still based on a system that we borrowed from bacteria, which is called the Cre-LoxP system. So this LoxP is a small sequence of DNA which has been identified in bacteriophagus, that are viruses that kill bacteria. So bacteria, to fight these viruses, they invented an enzyme that is called conditional recombinase, Cre, which actually can recognize these LoxP sites. So if you have a given X sequence between two LoxP sites, Cre recombinase will simply take it out and delete. However, and this is a way through which bacteria, they learned to fight this virus that they kill themselves. So when a bacteriophagus is infecting bacteria, bacteriophagus has LoxP sites, and bacteria with Cre is chopping its DNA. How we can translate this in mice? We start with a wild-type mouse. We are interested in this gene in this cell population. So we make a knock-in where we substitute this gene with another form of it that is flanked between two LoxP sites. And since LoxP sites are very small, this will not interfere with the normal expression of the gene. And on the other way, we can have a knock-in or a classical transgenic mouse that can have in this tissue of interest the expression of Cre recombinase. When we mate these two mice, we'll have a mouse that in cells where there is no Cre expression will maintain the normal expression of the X gene. Instead, where there is Cre, it will be a tissue-specific mutant. So the advantages are enormous because you can allow the precise targeting of different cell population. And also you can give temporal information of the role of a given gene, especially when you are dealing with lethal mutation. You can have naturally regulated promoters that can regulate key expression, like for example in adulthood. You can have inducible system. You can have some version of Cre that can be induced to function by injection of drugs. Or you can do local injection of viruses that express Cre. And on this I will come back later. And then the final proof so as I said now, we did a functional deletion of genes. But now there is also another way for the final proof of the role of a given gene, which is the functional rescue. So what normally people do for functional rescue, they do transgenic or viral expression of genes in their knockout animals. So you have a knockout animal everywhere. You want to re-express the gene in the hippocampus, you inject a virus or you make it transgenic. However, you have problems. The breeding are very complicated if you want to breed a knockout mouse with a transgenic one. The level of expression, especially with viruses, will be different from natural ones, and same thing, the pattern of expression. So how can you overcome these problems? You do what we do, the conditional rescue, which is a technique that was invented by Joel Elmquist in 2005. So what they did, they were interested in this gene. They made a knock-in in which, before the gene, they inserted a stop cassette flanked by two loxP sites. So this stop sequence will block all the transcription and the translation machinery. So you will have no expression of the gene of interest. So this will be like a knockout mouse, and you will have a certain phenotype. When you put Cre in a given particular cell type or with viruses, you will actually delete the stop cassette and trigger the expression of the gene. And if the mice are different, you can obtain a rescue of the phenotype from the knockout. And the good thing is that this rescue will be at the total wild type level, but only in specific cell types. And on this, so I explained to you how we can modify the genome of mice. But now there is an easy way to express protein, especially when you deal in the brain, which is what we call the viral vectors. So first thing people tried to do this was to use just nude DNA or DNA complex with lipid and polymer, which are very safe and not immunogenic. They're easily produced, but they can have toxicity. They cannot enter in cells. It's something that is not working anymore. So we need, if you want to put some genes in cells of mice without doing transgenesis, we need to use viruses. So how we can choose a viral vector system? We can have several families. We can have the retroviridea, we can have the adenovirus, and the parvovirus. So when you want to create a virus to express genes in mice, you need to choose what are your cell of targets, which can be dictated by the capsid of the virus, which is the external part. If there are cells dividing or not dividing, for example, if you want to target only dividing cells, you use retroviruses. If you wanted to target dividing and non-dividing cells, you can use both of these. If you want a transient or long-term expression, for example, the retroviridae, they are long-term expression. The adenovirus, they have very transient, strong expression. The parvovirus, like AV, they have very long-term expression. Also, however, the sizes of these three, they are not the same. AV are very small. So you also need to see how big is your gene. The immune response, because these two are very immunogenic. This one is less immunogenic. Whether the gene expression will be evaluated in vitro or in vivo, and also the safety, because for these viruses, you need access to a containment 2 lab. For this one, it's sufficient the containment 1. So this is just a table that you will find in the presentation that anyway I'm going to give it to you. And it can recapitulate what I'm saying, but What I want you to focus on is the main advantages of the 3 different type of viral vector that you can have with retroviruses, since integrates. You can have in oncogenesis, but often the good thing is that you have a persistent transfer. The adenovirus, they are very efficient in transfecting many tissues. However, the immune response of target cells is very high. And the best one so far are the adeno-associated viruses, because they're non-inflammatory, non-pathogenesis, and potentially long-lasting. However, be careful that the packaging size is much smaller. So this still is telling about the pathogenesis of original viruses, where you start with this mutated one. So this is something that you need to take into account. And this is still that you can see that AAV are the safest ones so far. So, of course, if you want to, as I told you before, with transgene, you can express genes in particular cell types. This you also can do with viruses sometimes. And this is called tropism. You can either target a large variety of cells or restricted cell types. And because not all vectors are designed to infect all cell types. So the tropism of the virus you're using will be the tropism of the vector you're using with transgenic mice. You can do this in retroviruses and adenoviruses that are pretty broad. AAV, they have broad tropism, but you can increase the tropism for a cell type with different serotypes. Or, as well, you can either use tissue-specific or cell type-specific promoters in AAV, or you can engineering different capsid serotype that you can have novel AAV hybrid vectors to broaden the leading tropism. But of course, what we are interested in is that you have a broad tropism vector, very safe, as I said you before, and you can restrict its expression to specific cell types. And this is a review with the protocol described how to make AAVs that I gave to you. And I just want to recapitulate the potentiality of AAVs now in neuroscience. You can have promoters that can be cell type specific, they can be ubiquitous, they can just be enhancers, or they can even have if you have a transgenic mouse with or another virus that is providing tetracycline, you can even drive the expression of this virus when you want with tetracycline system, as I explained before. You can express fluorescent proteins in viruses. You can express opsins for what is called optogenetic. We can talk about it later if you want. You can express DREAD. You can express sensors. You can express mRNA interference, or you can even use it with tetracycline as one. And you can even send this to different domains like nuclei. You can send to plasma membrane, presynaptic. For example, if you want to target some neurons, you can express with virus a GFP with the PDZ domain, which will send this GFP to the synaptic terminals. For my studies, I'm targeting a lot of protein to mitochondria. And this can become, the specificity of the promoter can be even enhanced by combining this expression to be constitutive, or to be injected in mice that can express CRE or flipase, which is another type of CRE, or even both of them sometimes to cross the two potentiality, in order to express in particular cell type according to the CRE mouse. And you can even repress the virus expression in particular cell type by using some target of microRNA that are specific for cell type. For example, you can inject the virus that will be silenced in neurons because you can have a mere particular sequence. And as you can see, you can have a lot of different cell type specific. I want you to focus mostly on this, that you can have synapsin promoter for neurons, GFAP for astrocytes, MBP for oligodendrocytes. You can easily target with TH promoter dopaminergic neurons, or GABAergic interneurons with DLX. And you also have, it's not listed it's not in this list, but you can also have CAM kinase 2 promoter to restrict viral expression to the glutamatergic neurons. But This is all explained in the article that I gave to you, where it's also the protocol. And as I was saying, the serotypes together with the specific promoters are very important because you can have different serotypes. The best one is characterized for the AV biology, it's the AV2. And a serotype is made by 60 capsid protein subunits, VP1, VP2, and VP3, that are arranged in a stoichiometric ratio of 1, 1, and 10. As you can see, when we take a virus and we lyse it and we put in a comacy gel, so it's a gel that is resolving the protein according to their molecular weight, we can see that we have VP1, VP2, and VP3, which is, of course, much more abundant. And the same thing if we blot an antibody against these three proteins. So the serotype, what they do possess particularly? They have some protein which they can interact with some receptors or with some co-receptors like heparan sulfate proteoglycan or fibroblast growth factor. These are all targets from the protein of the AV. So what will happen is that these AVs, they will bind to this receptor, and thanks to clathrin-mediated exocytosis, they will enter in the cell. When they are in the cell, they will, it's not complete the picture, but I can complete for you. When these viruses are in the cell, they will go into lysosomes, they will be destroyed, the DNA will be released, will go into the nucleus, and we start to express the gene. So normally, we can focus on the serotype that are normally used in the labs, that are normally used also together in neuroscience. We have AV1, AV2, and AV9. First thing, which was very used, was AV1 and AV2. Why? Because the transaction efficiency is much higher for AV1. Instead, intraparenchymal brain diffusion, when you do an injection or intramuscular or intrahepatic, is higher for AV1. It predominantly translates neurons, but AV1 can transduce glial ependydinal cells. So what we did something in the lab we started to do was to do an hybrid. So was to mix these two proteins and having a virus that has the property of both parental serotypes. However, this is not needed anymore because there is this magic AV9 now. Because as unique characteristic, it can cross the blood-brain barrier after intravenous injection. Intravascular can preferentially target non-lateral neurons and adult astrocytes, and is more readily transported in the brain compared to other serotypes. And after intracerebral injection, can mainly target neurons. So you can see that one serotype, according to the way injection, it can do what you want as a target. And if you combine this with the cell type specific promoter, you can really get, for example, the adult astrocytes or the newborn neurons. You can really infect what you want by playing with the serotypes and the capsids and specific promoters. So, as I told you, the gene delivery viral transduction starts with viral attachment to cell surface receptor or co-receptor, internalization, trafficking to the nucleus through endosomal compartment, escape from the late endosome, uncoating of the virion, conversion of the viral genome from single-strand DNA to double-strand DNA, and transduction after integration into the host genome. And playing with capsid and serotype, you can optimize tropism, diffusion of the virus, and transgene expression efficiency. So this was just to resume what I told you so far. But, of course, AAV, they exist normally in nature. But how we can artificially produce AAV vector? So, a non-natural AAV has the capsid, so it's a protein ensemble, which contains its genome that is made by two DNA sequences that are called inverted terminal repeats, and two genes that are called rep and cap that are needed to produce new virus. So what you can do to produce this virus is that you can have a viral DNA where you have between the two ITR your transgene. So here you have your promoter, your gene of interest, your protein signal, all what your tree-dependent site, all what you want. You have to put it between the two ETRs. And you can provide rep and cap genes with another plasmid. So if you do the co-transfection in what we call packaging cells with other auxiliary protein, you can generate an AV which will have the transgene you want. However, the auxiliary protein you need, it's very difficult. You can have different strategies. You can have stable lines of packaging cells. However, the stable integration of genomes of rep and cap it's very time consuming. And this, but however, this overcomes the limitation of transitory transfection methods. And this is not mostly used for large-scale preparation of AAV vectors that now are used in clinic. However, you still require the use of helpets virus. Why? Because this virus is called adeno-associated because it needs adenovirus to be produced. So this can limit the safety of adeno-associated adenoviral vector. And so all this, packaging cells, stable, and adenovirus, is mostly recommended for studies that require very high amounts of ARV, like preclinical studies in large animals or first step clinical studies that you can have huge amount of AAVs, and then there is a way to clean them from the adenovirus contamination. However, what we do in the lab and what we normally do for companies, for virus, for small amounts in neuroscience, is to use X cells. This is a normal cell line that expresses a lot of proteins with plasmid, very easy to transfect. And the particularity of this cell, since it was a tumoral cell, is that already express the protein E1 from adenovirus. So you have already one adenovirus protein that's provided by the cells. So what you can do is with the plasmid, given the E2 and the E4 at the protein of adenovirus, which we call the AD helper. So this cell will produce all the protein of adenovirus, but will not be capable of making an adenoviral particle. And if together with this plasmid, you give the RAB DNA that you want to pack, so with the 2ETR and the transgene, and the rep cap protein with another plasmid, what will happen? You will have the rep proteins, which will bind DTRs. You will have replication. The cap protein will be formed. The cells, thanks to the adenoviral gene, will assemble. And then from the cells, you can purify your AAV, which will be ready to go and completely clean of adenoviral contamination. So what you do to produce an adeno-associated virus, you do co-transfection of cells with the RAV plasmids, and this is the protocol we can discuss in detail if you want. You use packaging DNA plasmid and adenovirus helper plasmid, which does not contain the one which is provided by the X cells. It will be expression of all the construct and assembly in the X cells. Then you lyse X cells because adeno-associated virus, they are not released normally. So you lyse the X cells with hyperosmotic buffer, and this is to release the RAV particles. And then, since you have a total lyset, you don't want to have protein coming from the X cells, so you do purification and condensation of AAV particles. You can use two methods, either the heparin columns, because we know that some viral capsid, they can bind heparin. So you make them pass through heparin column, and the virus will be attached to the heparin, and the other protein will go away. But this is not suitable for every serotype. Or you can even reason that the viral particle, once you get rid of the EC nuclei, because you can easily get rid of the nuclei with a small centrifugation, a viral particle is heavier than protein cells because contains DNA. So what you can do what we call a density gradient, which is made by a sugar that is called iodixanol. So what you do, you prepare a gradient where you do an hyperosmotic phase, then you do 3 different phases of iodixanol at 3 different concentrations. So this is a gradient how it looks like, and you put this virus on top. And this, since iodixanol is acid, this color is due to a phenol red, which is a pH indicator, which can allow you to distinguish between the 2 phases. Why you do this? Because if you do an ultracentrifugation, you can see that in this gradient you will have formed a layer of protein waste and cellular debris. Here you have a white layers of empty particles. And then in this phase, because of the higher density, you will have the RV particles. So you just aspirate this phase, you concentrate, you dealyze the sugar with a phosphate physiological buffer, and you will have your RV purified. And with this, after you have been done this, you do a quality control of your viral preparation. So you do a Comassi gel to see that you have only viral protein and not other protein. And you do a titration, so you measure the physical amount of viral particles, because we know that each viral particle has one copy of DNA. So you do a quantitative PCR with primers that recognize the ETR sequences, and you can determine the number of effective particles to ml. And then once you have this, the only thing you have to do is inject this in mice and test it. And I think that I can finish here. I have, if you want to bring the discussion on the endocannabinoid system, we can go on it. But for what was my talk, I think we can finish here, and I already told you about transgenic and viral. And I think we can stop here, and I'm really open to get questions.